This video is going to be about proteins, one of our major macromolecules. Proteins are made of nitrogen, carbon, hydrogen, oxygen, and sulfur. So of and CHOPS, it's basically using all except phosphorus, okay? And then this will become an important part when we talk about how we understood that DNA was our genetic material, interestingly enough, even though we're talking about proteins. Proteins are made of monomers called amino acids. And this is a structure of an amino acid. All amino acids start off looking this way. There's a central carbon, an amine group, a carboxyl group, a hydrogen, and this R group. The R group on amino acids is a variable like X is in algebra. It makes each amino acid different. So everything else on the amino acid, everything else except for the R group, stays the same. The R group can change. And there are 20 different amino acids, and they differ in what their R group is. Here's a picture of 3 different amino acids. Notice again that that basic structure is the same in all 3 of them, but that the R group is different. Here we have an R group with different branching, short R groups, other branching ones, but with a charge, okay? And notice the table's labels here. Here we actually have categories of R groups based on the properties that it has. We can have hydrophobic, sort of non-polar R groups, or we can have hydrophilic, polar or charged R groups. And this is sort of an interesting thing. And the combination and order of the amino acids and what their R groups are will determine a lot about a protein. The polymer of amino acids, where they're going to be bonded together to make a polymer, is a polypeptide chain. Notice that prefix poly, meaning many or multiple peptides associated with proteins, or very frequently we will just call it a protein. Proteins are obviously used in our muscles. Most people know that. But it's also used to carry out all of our genes. So the reason that you might have brown hair is not just because your genes say so, but because your body has created brown color proteins. And the reason your eyes are a certain color is because of the proteins in them. And the reason that your body does a lot of its daily activities are because of proteins. Proteins are created by bonding 2 amino acids together to form a polypeptide. And just like carbohydrates had that special type of covalent bond called a glycosidic linkage, proteins have a peptide bond, which is the bond between 2 amino acids. And you do need to know the name of this bond. A protein can have 4 levels of structure, and you don't need to know this incredibly well. But there's primary, secondary, tertiary, and quaternary structure. So 1, 2, 3, and 4 levels of structure. And we obviously start at the primary and work our way up. Not all proteins will have quaternary structure. Primary structure is literally the order of the different types of amino acids. I have a methionine first, and a glycine, and a leucine, and an alanine, etc. It's like letters in words, A, B, C, D, etc. Just what are the basic orders of everything? Secondary structure allows sort of special folding. We create these things called alpha helices, so sort of a helix shape, and a beta pleated sheet, so sort of a folded sort of structure. Tertiary structure involves folding of these alpha helices and beta pleated sheets into larger 3-dimensional structures, so lots of beta pleated sheets and lots of helices together. Notice that we're continually zooming out. Here we see these, and these shapes make up only that sort of portion. And then last but not least, we have quaternary structure. Quaternary structure is where multiple proteins come together to form a larger unit. So this one tertiary structure comes together, and there might be several other different tertiary structures associated with that. So lots about creating this three-dimensional structure of proteins. And the thing you need to know about proteins is this major theme in biology is that form follows function. Or rather, structure is related to function. And so shape is directly related to its job. And if for some reason a protein's shape is altered, it can no longer do its job. Its shape is directly related to its job. And this loss of shape is going to be called denaturation, the loss of shape or unraveling of proteins, which causes them not to function properly. So proteins can be denatured by changes in the environment like salt concentration, temperature, and pH. And changes in even the primary structure, merely the little letters that make up words, just merely the order of things can have huge effects. For example, sickle cell anemia, which we will study again when we get to genetics as well as evolution, is only one wrong amino acid in a chain of hundreds, if not thousands, amino acids. And that changes normal red blood cells to look like sickles, and then they do not function properly. And this ends up not only being one wrong amino acid, but one wrong little letter in your DNA out of about 6 billion. And so again, you should be able to answer the following questions about proteins. And if you can't, please feel free to replay this video.